Let us discuss Biology Paper 1 for the KAPSABET High School Trial 1 exam. On to our first question. A young scientist observed a bird laying her eggs in a nest, and later the eggs hatched into chicks. Name 3 characteristics shown by the chicks that show a chick is a living thing but an egg is not. So we have quite a few characteristics to choose from. There is nutrition, movement, irritability. Number 2, which organelles should be abundant in Roman 1, skeletal muscle? Okay, so skeletal muscles require a lot of mitochondria. Now, reason being that these are muscles that contract to bring about movement. So every time a contraction occurs, energy in form of ATP is needed. This, of course, is produced by the mitochondria. Roman number 2, palisade tissue. This is where photosynthesis takes place. So the organelle that will be abundant here is the chloroplast. Question number 3. A Form 1 student was preparing temporary slides in the lab. In the course of preparation, he carried out the following processes. Okay, Roman number 1, sectioning, number 2, fixation, and number 3, staining. State the importance of the above processes. Okay, so let's start with sectioning. When it comes to sectioning, this, in simple terms, refers to the process whereby thin slices of a specimen are cut so as to be viewed under a microscope. Now, the reason why the sections have to be thin is so as to allow light to pass through. Remember, in a high school lab, we use the light microscope, which, depends on light for illumination of the specimen. Roman number 2, fixation. So fixation is a process that is undertaken during preparation of slides. So in this process, the specimen is treated with a solution that hardens it, and by doing so, maintains the structure of the specimen. Roman number 3, staining. So this is the process whereby you apply dyes to a specimen, okay? This is so as to ensure that the structures within the specimen become clear and distinct for viewing under a microscope. Question number 4. Why are lysosomes many in phagocytic cells? Okay, let's start with the term phagocytic cells. These are specialized cells that play a huge role in protecting us from pathogens. They are also generally known as white blood cells. Now you might be thinking, okay, then why not use the term white blood cells? Why refer to them as phagocytic cells? Simple, because not all white blood cells are phagocytic. Okay, let me break it down. Some white blood cells have this amazing ability of changing their shape. So this allows them to surround pathogens and then slowly digest them. Now this process is known as phagocytosis. Okay? Now to digest the pathogens, they need the lytic enzymes. These are powerful digestive enzymes, okay, that are found in lysosomes. So these enzymes break down any foreign materials into smaller, harmless substances that are then ingested by the phagocytes. Question number 5. Differentiate between guttation and transpiration. Now, guttation is the loss of water in form of water droplets. This occurs through openings called hydathodes that are normally found on the margins or on the edges of a leaf. Transpiration, on the other hand, is, yes, again, the loss of water, but in the form of vapor. This mostly occurs through openings called stomata. Question number 6. Give a reason why xylem vessel should be dead. Now the reason is simple, because being dead allows them to perform their main function, which is transportation of water. So this allows the water molecules to pass through the xylem vessels without being absorbed. because the cells are dead. Part B. What is the role of lignin in the wall of the xylem vessel? Now, lignin is a strengthening tissue, okay? It's there to provide mechanical support. Now, this is a complex carbohydrate and therefore very rigid. Question number 7. Name the disease of the blood characterized by part A, abnormally large number of white blood cells. Now, this disease is leukemia. So leukemia is a type of cancer that affects the bone marrow. And this is where blood cells such as red blood cells and white blood cells are produced. Now, the reason why leukemia happens is because of certain mutations that cause the bone marrow to keep on producing a lot of white blood cells. Problem is, a lot of these white blood cells are immature and not fully developed, and therefore cannot function properly. So their ability to fight pathogens is poor, making the individual to be more vulnerable to infections. Part B, crescent-shaped hemoglobin. Now, this is sickle cell anemia. So this is a blood disorder affecting the red blood cells. Now, in red blood cells, we have a pigment called hemoglobin. So in sickle cell anemia, the hemoglobin produced is abnormal, which causes the red blood cells to have a sickle-like shape as such, instead of the normal biconcave one. Question number 8. The chart below is a summary of a blood clotting mechanism in a man. So let's take a moment to look at the chart. Kindly pause the video if you require more time. Name the metal ion represented by Y. This, of course, is the calcium ion. Roman number 2, the end product of the mechanism represented by Z. This is going to be fibrin. Now, I am not going to discuss this in detail at this point. I've already done so in a previous video of mine. So I will link it below in case you want to watch it. And I highly recommend you do so. Moving on to question number 9. The graph below represents the growth of animals in a certain phylum. Study it and answer the questions that follow. Part A. Name the type of growth pattern shown on the graph. Okay. So this type of growth is known as intermittent growth. Part B. Identify the process represented by letter B. Letter B represents molting, also known as ecdysis. Part C. Name the hormone responsible for the process in B above. The hormone is known as ecdysone. This, of course, derives its name from ecdysis, from the process of molting. Okay, now let me discuss this, right? So, intermittent growth is a type of growth that is seen in arthropods, insects, crustaceans, and so on. These animals have an exoskeleton, a very rigid, hard external skeleton that is made up of a complex carbohydrate called chitin. Now, this exoskeleton is very important. It provides structure and protection to the animal, but it doesn't stretch as the animal grows. So as you can imagine, the arthropods cannot grow continuously like other animals that have an internal skeleton. So they undergo molting, where they shed their exoskeleton, grow and expand in size, and then finally, develop a new, harder exoskeleton. So this process repeats several times until the arthropod reaches maturity. Now, this process is controlled by hormones. So there are 2 hormones that are involved. We have ecdysone hormone, as stated, and another hormone called juvenile hormone. Moving on to question number 10. Explain why a mule is infertile. Now, a mule is an offspring of a donkey and a horse. So these are 2 animals that belong to 2 different species. Therefore, they cannot produce a fertile offspring. Question number 11. Phylum Arthropoda is the most successful of invertebrates. Explain two characteristics that make them the most successful. Okay, so number one, they have a hard exoskeleton made of chitin, which protects them from predation, that is from predators, and it also protects them from desiccation. Desiccation is the extreme loss of water. Number two is that they also have jointed appendages that are adapted for different functions. Moving on to question number 12. Name the phylum whose members possess a notochord. This, of course, is going to be phylum Chordata. Question number 13a. Define evolution and homologous structures. So evolution is the gradual change of living organisms from simple life forms to more complex life forms over a very long period of time. Homologous structures are structures with a common embryonic origin, but have been modified to perform different functions. Part B. State 3 limitations of using fossil records as an evidence that supports organic evolution. Okay, so number one is that you're going to have missing links, gaps, because some fossils have not yet been discovered. Number two is that you're going to have destruction of some fossils. This is due to certain activities like earthquakes. And three, the soft-bodied parts will decay, okay? So you're going to end up having an incomplete picture. Question number 14. The following is a part of a kidney nephron. A, Roman 1, name the process represented by the arrows. Okay, so this is our nephron. Okay, that is the nephron the way it is. It's the basic functional unit of the kidney. Now, the two parts represented in this diagram are the glomerulus and the Bowman's capsule. The process that is taking place here is known as ultrafiltration. Now, this is a process whereby blood is filtered under very high pressure through tiny pores that are present in the endothelium of the glomerulus. Okay? Roman number 2. Name the conditions necessary for the process named in A, Roman 1 above to take place. Now, from the definition I just gave, I bet you can pick out the 2 points I mentioned. So number 1 is you need to have a very high pressure. And number 2 is the presence of pores in the endothelium of the glomeruli. Now moving on to part B, identify with a reason vessel A. So vessel A is the afferent arteriole. Okay, the reason is it has, because of the direction of the blood flow. Now I'm going to tell you this. We have the afferent arteriole and the efferent arteriole. Now there are 2 ways usually that you can differentiate between the two in diagrams. Number 1 is the direction of the blood flow. So the afferent arteriole transports blood into the glomerulus. So A is the afferent arteriole. The efferent arteriole away from the glomerulus. The other is based on the width of the lumen of A and B. Now the afferent arteriole, which is A, is supposed to be wider than the efferent arteriole. But I'll be honest with you. I cannot tell from this diagram that there's a difference in the width between the two. They just look the same to me, okay? If you can spot a difference, in the width of A and B, kindly let us know in the comment section. So as of now, we are going to stick with the direction of the blood flow. Part C, name any 2 blood components that are present in vessel A but absent in vessel B. Okay, so we have urea, we have glucose, amino acids, salts. So urea is a waste product. It's going to pass through the nephron and finally be transported to the bladder for removal in urine. Glucose, amino acids, and salts are going to be reabsorbed. These are useful products that are needed by the body. Now, I would like to say this. Excretion, especially, the part involving the nephron, is one that I have yet to cover. And I'm still astounded that I have yet to cover it, considering that it's one of the more commonly tested areas. But I want to tell you this. I am planning on discussing it in detail soon. So if this is a challenging concept for you, stay tuned. Question number 15. The diagrammatic representation below illustrates one of the processes that occurs in mammals during feeding. Carefully study it and answer the questions that follow. Okay, so we have the gullet. So the gullet is another name for the esophagus. And we have a bolus, okay, actually two boluses there moving across. So Roman number 1, identify the process. This, of course, is peristalsis. Roman number 2, state 2 structural adaptations of the gullet to its functions. So we need to come up with adaptations of the esophagus. What is the function of the esophagus? It's a tube-like structure that provides passage for the food from the mouth into the stomach. So adaptations. Number 1, it has circular and longitudinal muscles that contract and relax rhythmically in order to push food along. Number 2, it has, an epithelial lining that has goblet cells. Now, goblet cells are specialized cells that are found along the alimentary canal. These cells secrete mucus. Now, the mucus produced lubricates the food, which reduces friction and therefore makes the movement smooth. Okay, Roman number 3, name one enzyme already present in the food bolus within the gullet in man. Now let's just go back a bit. The food starts out in the mouth and then proceeds to the gullet, okay? So if you're talking about an enzyme that is present in the bolus, so that means that this was an enzyme that, was mixed in with the food in the mouth. Remember, the esophagus does not secrete any enzyme. So which enzyme is present in the mouth, specifically in saliva? That is enzyme salivary amylase, also known as ptyalin. Part B, state 2 functions of mucus secreted by the intestines. So as mentioned, goblet cells which secrete mucus are found all over the alimentary canal. Now they have several functions. So let's mention 2. Number 1 is that they lubricate the food, reducing friction and making movement smooth. Number 2 is that they also prevent autodigestion of the lining of the intestines by the protein digesting enzymes. Question number 16. Explain each of the following. Part A. Variegated plants accumulate less food than non-variegated plants under similar conditions. Okay, so variegated plants contain variegated leaves. These are leaves that have parts that are green because they contain chlorophyll, and other patches that are either white or yellow. These patches lack chlorophyll. So as you can imagine, variegated leaves are going to have less chlorophyll, therefore they are going to produce less food through photosynthesis. Non-variegated plants, on the other hand, have leaves that are entirely green. So they have more chlorophyll and therefore will synthesize more food. Part B. Most leaves are thin with a broad leaf surface. Okay, so we're being asked to explain 2 things over here. Number 1, the reasons why leaves are thin, and number 2, why they are broad. Now, when it comes to the thinness of leaves, it reduces distance for diffusion of carbon dioxide, which is needed for photosynthesis. Leaves are also broad in order to provide a large surface area for maximum absorption of light and carbon dioxide for photosynthesis. Question number 17. Set the economic importance of the following plant excretory products. Part A, we have papain. Okay, so papain is used as a food tenderizer. B, caffeine, it's a mild stimulant. Part C, colchicine. This is used in cancer therapy. Question number 18A. State two processes which occur during the anaphase of mitosis. So we have separation of the sister chromatids, and number two, the sister chromatids also move to opposite poles of the cell. Part B. What is the significance of the first meiotic division? Now remember, meiosis occurs in two phases. We have the first stage and the second stage. So the first meiotic division brings about separation of homologous chromosomes. In case you're wondering about the second meiotic division, that brings about separation of the sister chromatids. Now, part C set two ways in which HIV/AIDS is transmitted from mother to child. Number one, during birth, and number two, through breastfeeding. Number 19, state the function of the following during pregnancy. Part A, the amnion. So the amnion secretes the amniotic fluid. Part B, the amniotic fluid, this acts as a shock absorber against mechanical injury. Part C, the umbilical cord. This connects the embryo and the mother to allow for exchange of substances. Question number 20. Name the process by which producers convert sunlight energy into chemical energy. This, of course, is photosynthesis, where light energy is converted into chemical energy. Roman number 2. Chemical energy is converted into heat energy by consumers. This is respiration, whereby food substances that contain the chemical energy, are broken down in order to release energy with a little bit of heat. Question number 21. Students from the M-Pesa Foundation Academy wanted to investigate the population of crabs in their school pond. They caught 50 crabs, marked them with white paint on the cephalothorax, and then released them back into the pond. After 3 days, they came back and caught 50 crabs, of which 3 had the white mark. Using the data above, calculate the population of crabs in the pond. Okay, so we're going to use the following formula. First marked multiplied by the second captured over marked recaptured. So how many crabs were first collected and marked? 50. How many were captured in the second lot? Again, 50. How many were captured in the first lot as well as the second one? Only 3. So this will give us 833.3. Now we're talking about crabs, organisms, so we can't have decimal places, okay? So it's going to be 833 crabs. Part B, suggest 3 assumptions the students made during this study. Okay, assumption number 1 is that the marked crabs freely interacted with other organisms. Number 2, there was no entry or exit of crabs during this study period. Number 3 is that the mark given to the crab did not affect their behavior. Question number 23, part A. State how the following factors increase the rate of diffusion. Okay, factor number 1, temperature. So an increase in temperature increases the kinetic energy of particles, which causes them to move faster, therefore diffuse faster. Roman number 2, the diffusion gradient. So the diffusion gradient refers to the difference in the concentration of particles between 2 regions. So the higher the diffusion gradient between 2 regions, the faster the rate of diffusion. Roman number 3, the size of diffusing particles. Okay, so the smaller particles move faster, therefore increasing the rate of diffusion. Question 24. Waterlogging in terrestrial plants inhibit uptake of certain mineral ions from the soil by the plants. Explain. Okay, so terrestrial plants are plants that are found on land. Now, waterlogging refers to the condition whereby the soil becomes so saturated with water such that even the spaces in the soil particles that would normally be occupied with air would instead be filled with water. So as a result, the concentration of oxygen in the soil is lowered. It becomes low. So this is going to interfere with the active absorption of some mineral salts. Now, some mineral salts are going to be absorbed by active transport. Some are going to be absorbed by diffusion and some by active transport. Now, as stated, diffusion is a passive process, okay? So it will happen regardless. Active transport, on the other hand, requires an input of energy. And how is energy produced? Energy is produced during respiration. And respiration requires oxygen. Do you see how all these tie together? If you have a low concentration of oxygen, respiration cannot take place efficiently. Energy is not produced. Therefore, active absorption of mineral ions is going to be inhibited. Part B. Set 2 illustrations of osmosis in plants. So we are simply being asked to give examples of osmosis taking place in plants. So we have closing and opening of stomata, feeding in insectivorous plants, absorption of water from the soil, and many more. Question 25. The diagram below represents a gill of a fish. Take your time and go through the diagram. Roman number 1. State 2 ways in which a large surface area is created in structures labeled K. Now, K refers to gill filaments. This is where gaseous exchange takes place. Now, for gaseous exchange to take place, we need to have a large surface area. So how is a large surface area obtained when it comes to gill filaments? In 2 ways. Number 1 is that they are numerous, they are quite many, as you can see. And number 2 is that they are long. So both of these ensure that we have a large surface area for gaseous exchange to take place in the gills of a fish. Roman number 2. Name the type of flow system that occurs between water and blood in the capillaries present on structures K. Okay, so this is our countercurrent flow system, whereby you're going to have 2 liquids or solutions moving in opposite directions. This maximizes the movement of the respiratory gases, that is oxygen and carbon dioxide. Roman number 3. Name an organ in human beings that also displays the flow system named in 2 above. Okay, so the organ is the kidney. Now within the kidney, we have the nephrons. Now, in the loop of Henle within the nephron, you're going to have a counter-current system as such. But you cannot state your answer as the nephron because the question is asking you for an organ, and that is the kidney. Question 26, which is our last question. Identical twins were separated after birth and were then raised in different environments. One in Kenya and the other in USA. They rejoined after 18 years and it looked slightly different. Roman 1. Name the type of variation the twins exhibited. This is going to be continuous variation. Now the reason is because discontinuous variation is not affected by the environment. Okay? Yeah. So continuous variation. Roman number 2. Give 2 observable differences likely to be noted within the twin. So we're going to give differences that might be present between the 2 and which will fall under continuous variation. So skin color, height, body weight. Okay, any of these is correct. And that, ladies and gentlemen, brings us to the end of this amazing marathon of revision. See you next time.